Okay, good morning, everyone. We're going to get things started here in an attempt to try and stay on time. Welcome to this morning's session. This is session 5, Stem Cells Regeneration in Neuromuscular Disease. My name is Gianni Parise from McMaster University, and I'm chairing today's session. I'll also be your first speaker this morning. Before we get started, I'd like to thank Dr. Hood and the organizing committee for inviting this symposium. I think we have a great lineup of speakers this morning. Most of the great comes after I speak, and I'll be followed up by Dr. Charlotte Peterson from the University of Kentucky, who will be speaking on the potential role of satellite cells in exercise response heterogeneity. Dr. Douglas Millay from Cincinnati Children's Hospital Medical Center, who will be speaking on multinucleation and myonuclear accretion during muscle development and adaptations. And Dr. Marnie Bopart from the University of Illinois at Urbana-Champaign, who will be speaking on development of extracellular vesicle strategies to recover muscle after disuse. I'm going to start us off talking about the relationship between muscle stem cells and capillaries. And I have to be honest with you, I'm kind of tired of talking about this. I was hoping that we would have wrapped this story up a few years ago, and we probably would have, but we're, like everyone in the room, delayed on our research due to the pandemic. And so the final piece of this story, I think, is ready for presentation. So I'm going to be speaking about this a little more today. I'm going to be speaking about it, from the basic cellular concepts and some of the basic observations we've made over the years to some applied strategies that we've attempted over the last couple of years. The reason why we're interested in the relationship between satellite cells and capillaries is because we believe satellite cells and capillaries, or we believe that capillaries play an important role in regulating satellite cells. And when we look at the function of stem cells or satellite cells in skeletal muscle, I think we're at a point where we can appreciate that they do a lot of things, right? And so understanding how they're regulated is critically important. But what do they do exactly? And I think there's absolutely no dispute that satellite cells are critical for skeletal muscle regeneration. In fact, Dr. Peterson's group has shown quite nicely that regeneration does not happen in the absence of satellite cells. So that's an obvious function of satellite cells. There's evidence that satellite cells are important for growth, all right? Probably one of the more contentious issues, but I think there's now a body of literature that would support that satellite cells do contribute to skeletal muscle hypertrophy in response to resistance exercise. And of course, satellite cells are involved in non-hypertrophic adaptations, so the types of remodeling of skeletal muscle that are associated with aerobic exercise. And finally, in another discovery from Dr. Peterson and her collaborators was the idea that satellite cells are important vehicles for signaling. And we've seen over the last few years some really elegant work demonstrating quite nicely that satellite cells can produce and package exosomes and regulate various aspects of skeletal muscle repair. All this to say that understanding how these functions occur, in my opinion, is interesting, but also critically important. And when we think about satellite cell regulation, we can all appreciate that satellite cell regulation is really quite complex. And this is actually a figure that was taken from a paper done back in 2010 by Bensinger and colleagues, where they show you here a single fiber and a satellite cell residing on that fiber. And signals can come from a number of sources, including the fiber in which it resides, adjacent fibers that might be undergoing regeneration or remodeling, other cells in the skeletal muscle milieu, such as inflammatory cells, like macrophages. Fibroblasts can contribute signals. And we believe that the local circulation in skeletal muscle is a rich source of signal, okay? And so we wanted to explore this further. And it should be no surprise, right, that capillaries might be an important player in the regulation of satellite cells. This is an image that's compliments of Oliver Baum, who is an author on this paper, this review. And what this is a mouse muscle that you're looking at, and you're seeing an artery terminate. It's running perpendicular to the muscle fibers. You can see it terminate and give rise to capillaries, and these capillaries are tortuous along those fibers, allowing for efficient exchange, of course, of oxygen and nutrients and delivery of CO2 and metabolites away from the skeletal muscle. And so there's already this intimate anatomical relationship that exists between muscle fibers and capillaries. And if you take this and we flip it on its side and look at a cross-section, this is an electron micrograph. You see 3 fibers here. Right in the center, you see a capillary, and you can see that actually an erythrocyte's been captured in this image. And right to the side here, in very close physical association with the capillary, is a satellite cell. Now, we weren't the first people to start to explore this relationship, and in fact, the first paper exploring satellite cells and capillaries was published by Christoph and colleagues back in 2007. And it was a really simple study. What they did was they looked at a number of electron micrographs and just identified the structures. And what you can see here in this image is a capillary and an endothelial cell. And just outside that, we can see a satellite cell in very close proximity. In this image here, you see a satellite cell down here, and you see a capillary right over here. And you can see this physical projection from the satellite cell out towards that capillary. And their conclusion at the end of this is that satellite cells are largely juxtavascular, and that, of course, this must be important in the function of satellite cells. They did go further, and they looked at activation status and showed that, in fact, when you co-stain satellite cells with MIF5, you can see that these satellite cells, in close approximation to the capillaries, are, in fact, active. So we decided to take this a step further and explore this relationship a little further, and this was done primarily at the time by my PhD student, who's now a postdoc, Dr. Josh Netterveen. You might have seen him around. He looks a little different. He's had a child since then, so there's more gray hair and a bigger beard, but nonetheless, he's pretty much the same. And Josh painstakingly actually measured the distances between satellite cells on a fiber and its closest capillary. And what he found, in fact, is that there's a range of distances, which wasn't a surprise, of course. And when he looked further and wanted to try and understand this relationship a little more, he actually stained for the activation status of satellite cells. And what he found was really quite interesting. And that is that satellite cells that were in the active state, that is, that they were expressing MyoD, were about 20% closer to their nearest capillary than their quiescent counterparts. Okay, so this is distance to a capillary, and these are quiescent cells, and these are active cells. And this simple observation led us to believe that, in fact, whatever is coming from those capillaries is important in activating satellite cells. So we wanted to explore this further yet, the idea being that we developed a hypothesis that basically if a satellite cell is closer to a capillary, it has the ability to activate when it interacts with the signals that are being released from the capillaries, right? So we developed a hypothesis that we refer to as the capillary impact zone. The idea being that the concentration of signal being released at the site of the capillary would be greatest nearest the capillary and could activate cells. As those signals diffuse away from the capillary, the concentration drops, and perhaps satellite cells can't respond in the same way. And this is relevant in cases like aging. So here we're looking at a number of capillaries, and you can see this green indicates signal being released from the capillaries. And as the signal diffuses away from the capillary, the concentration gradient drops, and that satellite cell may not be able to respond. And so this was the hypothesis that we were working with, and we want to explore this further and try and understand whether or not being closer to a capillary would result in greater activation or more efficient activation of satellite cells and expansion of the satellite cell population. So the idea was individuals who have more capillaries will have a more efficient response to damage than those who have fewer. And Dr. Netterveen ran a study where we had recruited about 30 individuals, and we collected muscle biopsies from them at baseline. We then damaged them with 300 eccentric contractions, and we took biopsies subsequently at 6 hours, 24 hours, 72, and 96 hours. And we looked at the response of the satellite cell population over that time course. Importantly, after recruiting these individuals, we grouped them into a group that we referred to as a high CFPE group, and CFPE stands for capillary per fiber perimeter exchange ratio. All this is a proxy measure of perfusion. Okay, so we refer to it, and I'll refer to it this way from now on, a group with the highest capacity for perfusion and a group with the lowest capacity for perfusion. Okay, And if our hypothesis is true, then we would expect a better response in individuals, a better satellite cell response in individuals in the individuals from the high capacity for perfusion group. First thing we looked at was activation of the satellite cell population. We did that by co-staining with MyoD. And it's hard to see here on this here. You'll have to trust me when I tell you that we have a PAX7 positive cell here co-localized with MyoD. But when we looked at activation status, it's essentially exactly what we thought we would see, that individuals, and the black bar here represents individuals with the highest capacity for perfusion. So they have more capillaries, and the open bars represent individuals with the lowest capacity for perfusion. And what you can see here is that you see a rapid activation in the individuals with the highest capacity for perfusion, and it's sustained through 72 hours. And we see a blunted activation status in the individuals with the lowest capacity for perfusion, suggesting that capillary content and the signals that are being released from those capillaries might be important for the activation of satellite cells. So we want to look at expansion of the satellite cell population, and it stands to reason that if you're more efficiently activating these cells, that they would expand to a greater degree. And in fact, when we look at satellite cell number here, across the time course, again, black bars representing those with the highest capacity for perfusion, you can see a rapid expansion peaking at 24 hours, sustained through 72 hours, whereas over the first 24 hours, in the people with the lowest capacity for perfusion, fewer capillaries, you see that the response is quite flat, okay, suggesting to us that the presence of those capillaries is critically important for the satellite cell response. We did look at this in a fiber type specific manner as well. And when we looked at the type 2 satellite cell pool, and you'll see why I think this is important in a moment, you see that the response is even exaggerated further, right? So again, if we're looking here at expansion of satellite cells associated with type 2 fibers, again, you see this rapid and sustained expansion of satellite cells over the 72-hour time course. And again, almost an entirely flat response in individuals with the low capacity for perfusion, okay? Why this is important, of course, is that we can appreciate now that the fiber type that is particularly affected by things like aging tend to be type 2 fibers, right? And we did a study where we actually investigated the relationship between capillaries and satellite cells in older adults, and what you're looking at here is simply capillaries per fiber, and then the capacity for perfusion over here. But on the left, what you'll notice is type 1 and type 2 fibers, young represented by the black bars, the open bars representing older individuals. And you'll notice that in the type 2 fibers, we're seeing fewer capillaries. And of course, that translates to less capacity for perfusion. If we look at the number of satellite cells in older individuals, again, type 1 and type 2, again, you'll notice that we have fewer satellite cells in older adults as compared to younger adults. And if you put those two things together, where you have fewer capillaries and then fewer satellite cells, the last thing to appreciate is that the distance between satellite cells and capillaries is greater in older individuals, which would suggest that they're more compromised in activating satellite cells, which is what we and others have shown multiple times now. Now, Dr. Tim Snyders, who's also here, and his colleagues, and of course my colleagues, Luc Van Loon, ran a resistance exercise training study. It was a 24-week study, pardon me. It was a 24-week study where they took older individuals and they took baseline biopsies, biopsied them again at 12 weeks and again at 24 weeks. And again, splitting the groups up into a high capacity for perfusion group and a low capacity perfusion group. And found some very interesting things. The first was that if you look at the type 1 fibers, really, we don't see a whole lot going on. So just to orient you, we're looking at low capacity for perfusion, high capacity for perfusion, and this is fiber size or fiber cross-sectional area. In neither the low or high group do we see a significant increase in fiber cross-sectional area, although it's worth pointing out that you do see an increase at 12 weeks in the high group, it just doesn't reach statistical significance. However, if we look at the type 2 fibers, right, again, in the low group, through 12 weeks, we see no change in muscle fiber size. We do see it increasing at 24 weeks, a fair bit of variability and no statistical significance. But have a look at the high capacity for perfusion group, right? Substantial increase in fiber cross-sectional area at 12 weeks, even higher at 24 weeks. And these are the individuals who have a higher capacity for capillary perfusion. If we look at the satellite cell content, again, type 1 fibers, not a lot going on in the low capacity for perfusion group, but in the high capacity, you can see an increase at 12 weeks. It's sustained through 24. Really where the exciting stuff is, though is if in the type 2 fibers. And if you have a look here in the low capacity group, not a whole lot going on, nothing going on at 12 weeks, maybe a blip at 24 weeks. But the high group, pardon me, you can see that those who had a high capacity for perfusion had the ability to increase satellite cell content substantially at 12 weeks, and that was sustained through 24 weeks. Okay, so this is just further evidence that the relationship between satellite cells and capillaries may be very important, particularly in aging. So the last piece of the puzzle for us was trying to understand whether or not we can manipulate these factors and develop or optimize a strategy for increasing muscle cross-sectional area based on everything we've learned about the relationship between capillaries and satellite cells. And so what we did was we ran a training study. This was a unilateral leg training study where individuals came in and we took biopsies from them. We then randomized their legs so that one leg underwent 6 weeks of aerobic conditioning 3 times per week, while the other leg remained sedentary. Then both legs went into a period of resistance exercise training. The idea here being that we wanted to try and increase capillary content in the one limb relative to the other, then have them perform resistance training for 10 weeks and see if we would end up with better outcomes in the group that was aerobic, or in the limb that was aerobically conditioned. So we took biopsies at baseline following the aerobic conditioning period and again following the resistance exercise training period. You can see here that we did single leg VO2 max at the start as well, to give us a sense of whether or not we were seeing adaptation following that aerobic conditioning period. Okay, so what did we see? First thing to point out here is that the model worked quite well, right? If we look here, what we're looking at is baseline pulmonary VO2 in both the trained and untrained leg. And then what we're looking at here is the post-aerobic conditioning period. This is really the only thing that matters for this slide. And what you can see here is in the black dots, we're looking at the group, or the limb, sorry, that was aerobically conditioned. In open dots, we see the limb that was not. And so you can see there was an increase in single leg pulmonary VO2. There was also an increase in a single leg VO2 work peak in the aerobic conditioned leg versus the non-aerobically conditioned leg. So we did achieve the goal of increasing VO2 in that limb. When we look at capacity for perfusion in both limbs, again, what you'll see here is this is CFPE, or capacity for perfusion. You can see that in the non-trained limb, there's no significant increase. However, in the type 1 fibers and the type 2 fibers, frankly, we can see that there is an increase in capacity for perfusion in the leg that was aerobically conditioned. So now we have individuals going into the resistance training phase of the study where we know that one limb has a greater capacity for perfusion than the other. And what does this mean for the resistance exercise training period? We have a lot of data. I'm just showing you some key pieces here in the interest of time. Importantly, what we're looking at here is cross-sectional area of type 1 and type 2 fibers on the right. And what you can see here in the open bars is the resistance trained limb that did not go through aerobic conditioning. And you see absolutely no change in fiber cross-sectional area in the type 1 fibers, whereas we do see a significant increase in the limb that was aerobically conditioned before resistance exercise training. We see the same thing in type 2 fibers. Now, the difference, of course, in type 2 fibers, we do see an increase in fiber cross-sectional area in the non-aerobically conditioned limb, but it does not reach significance, whereas we see a significant increase in fiber cross-sectional area, type 2 fiber cross-sectional area in the aerobically conditioned limb, suggesting that, in fact, this is something that we can manipulate. We can develop a strategy that might result in more optimal gains in muscle fiber cross-sectional area. There are a couple other observations I wanted to show you. Sorry, this is, yeah, a little slow. I don't know why. This slide is delayed every time I hit a button on it. But anyway, we looked at our data following all the analysis, and we broke out our data here looking at, in this case, we went with high and low responders with respect to with respect to capacity for perfusion. So in the black bars, we have individuals who had the limb that had the highest capacity for perfusion post-aerobic conditioning versus the limb that had the lowest capacity for perfusion post-aerobic conditioning. And what you can see here on cross-sectional area is that in both the type 1 and type 2 fibers, the greatest increases in cross-sectional area happened in individuals who had the greatest capacity for perfusion at the end of aerobic conditioning, okay? And on the right here, we looked at high and low responders with respect to changes in cross-sectional area. What you can see here is that in the black dots are individuals who have the greatest increases in cross-sectional area in type 1 fibers here and type 2 fibers here. And what we show here is that those individuals with the greatest increases in cross-sectional area also had the greatest change in satellite cell number over the resistance exercise training period. Okay, so the individuals who had the biggest gains in cross-sectional area through resistance training also had the biggest gains in satellite cell number. And I'm not sure that Dr. Peterson's going to be speaking to heterogeneity in response to exercise and satellite cells. And we haven't talked about this, but I hope we're on the same page. Nonetheless, just to summarize quickly, the anatomical relationship between capillaries and satellite cells is functionally important. I think we've shown that in multiple ways. The relationship can be manipulated through exercise to improve training responses. And the next logical question, and I'm not sure I have the appetite to do it, is can this strategy be utilized therapeutically to improve the exercise response in other populations, such as older adults? With that, I'll just acknowledge my current lab group here. These are the individuals in red who contributed significantly to the data I just showed you, and of course, my collaborators and funding sources. And I'm happy to take any questions.
It's really fun to see the story all come full circle here. Very nice. I wanted, speaking of response heterogeneity, I noticed in your group that trained aerobically and then resistance trained, suddenly the increase in fiber, or the change in fiber cross-sectional area in that group in particular, the variability was huge, much higher than in any other groups. Did you do any kinds of correlations to see if that was, that variability was correlated, say, to satellite cell content or capillaries, or capillaries, being from the US, capillaries, so that to try to understand that heterogeneity?
Yeah, we did a ton of correlations, honestly. I mean, I can't keep them all straight. In the end, nothing was coming out to explain the variability. Okay. Again, statistically, things filtered out in the way that we expected them to, but yeah, I don't think we have a correlation that would explain it all. Thanks. Karen?
Just beautiful. And the design on that last study was just mind-boggling to me.
It was the biggest pain.
I'm just trying to imagine being that subject.
Yeah.
But I actually have a follow-up to what Shar just said, because you've got so what's beautiful about it is you have the same human, and you have samples that have different experiences in the legs. So I'm assuming you did some statistics within, so it seems statistically more powerful to actually compare within one leg to the other. And I'm seeing you smile, so this is obviously something so can you share anything that you learned doing it that way?
Oh, so.
Sorry.
So at the start of the study, I thought that this was a really clever way to do this. And,
it is.
And I think, physiologically, it's great. Statistically, this was the biggest pain ever. My student, Aaron Thomas, who ran this study, and his committee, we might have discussed statistically how to analyze this for 10 hours. It was just, and so it was such a pain for me that I pledged to never run a study like this again. But physiologically, I love it. So, I mean, we could probably talk about the optimal way to statistically analyze things all day long.
Yeah, and I wouldn't be that person, but there are those people out there. So
absolutely,
I'm just
absolutely,
saying.
Yeah.
It's amazing.
Thanks, Karen. Appreciate that.
Very nice talk. I just want to be a bit provocative because I'm a vascular biologist and I look at the muscle from a vascular-centric point of view. So my question is, can you somewhat hypothesize or maybe give me your thoughts on how sure you are that this is a satellite cell mediated observation, and might not be or might be directly mediated via the vasculature itself. I mean,
Yeah.
I'm sure you are aware of the recent Nature Metabolism paper, which is a bit difficult from my point of view, where actually the authors showed that there is direct signaling from blood vessels to the muscle.
Yeah, it's a great question. And I think it's really important to recognize right out of the gate that this was aerobic conditioning that I've been referring to, and I've been focused on the capillaries. In fact, I had the opposite view that you just had, and that is that it may not be the capillaries per se. There's lots of things that happen when you aerobically condition muscle. It could be something else. I think it's hard. There's so many associations that we're making here between the capillaries and satellite cells that it'd be hard to convince me that it's something else that's responsible for the bulk of what we're observing. Your point's well taken, and I'm not really sure how to answer it. There are lots of other, we could talk about parasites as well, and whether or not there's something going on there. But I appreciate you highlighting the point. I just don't have an answer for you. Thanks. Hi.
Hi, I'm Fran from University of Arkansas, and I was just wondering if there's anything out there about biological sex differences in perfusion, and then what that would mean for muscle regeneration differences in males and females, and the response to exercise in general, if you can speculate on that.
So, I mean, it's a great question. I think when you look back historically at some of the regeneration data and repair data, there was some sex-based stuff that was done in mice years ago, and there's been this lingering idea that perhaps estrogen has protective effects against damage, and that would, of course, impact the repair process. I don't think enough work's been done, frankly, to say whether or not that those are bona fide facts at this point. We're actually embarking on a huge sex-based damage and repair study right now. We're in the middle of the trial now, actually. And so I suspect we'll have answers for you over the next year. I think at this point, I mean, one of the things that we did make an observation, I'm not sure, we haven't put it out there yet, we did make an observation that when we damage women compared to men, we see a completely absent satellite cell response at the same point where men are starting to spike in satellite cell number, which is what led to this big trial that we're doing now, is that, maybe we need to go back to just some simple damage studies and identify whether or not, for the same work, we experience the same level of damage in men and women. So there's lots of work to do there. Thanks. Okay, with that, I will pass the baton to Dr. Charlotte Peterson for her talk.